Good evening, Nita. Good afternoon. Can you hear me, Nita? Nita?
Yes, sir. Yes, sir.
Okay. Yeah. I'll keep that this thing ready for share also. Okay.
Yes, sir.
If there is some. Good afternoon, Sheikh bhai. कैसे हो? Speaker has come, Dr. Mohammed Sheikh. कैसे हो सर? शेख भाई. Good afternoon.
Hello, good afternoon, Pramod ji.
Good afternoon, Sheikh भाई. कैसे हो? बहुत सालों बाद, 15 साल बाद. Life. 15 years. Almost, yeah. 2007.
हाँ 2007 15 साल हो गए प्रमोद जी.
15 साल के बाद देखेंगे लगता हम एक दूसरे को. After 15 years.
एक 2 मिनट में ज्वाइन करता हूँ.
Okay, शेख भाई.
म्यूट। ये क्या होगा?
नीता मेरी स्टूडेंट है, थर्ड ईयर B.Pharm की। She looks after most of this webinar programs. वो- वो खड़ में किसी को बुलाया शेख भाई आपने? मैंने मैसेज तो डाला है।
हेलो।
हाँ शेख भाई, वो खड़ में से
हाँ
किसी को
प्रमोद।
अपना
हाँ डाला है मैंने,
शिवाजी
ग्रुप
को
पे डाला है।
शिवाजी को मैं डालता हूँ एक बार।
हाँ शिवाजी को डाल दीजिए।
पवार। येवले सर कैसे? पाटिल सर को भी एक बार डालता हूँ।
हेलो।
हाँ बोलिए।
सब लोग अच्छे?
हाँ, बढ़िया। बहुत साल के बाद।
साल हो गया, आप भी औरंगाबाद नहीं आए।
मैं आके गया था एक बार, तब मैं आया था, कंपनी आके आया था, मिले नहीं, कुछ लोग नहीं मिले। तभी दंगे
ओके
हो गए थे ना, हम आने का टाइम दंगे हो
ओके
गए
ओके।
थे, तो मैं नहीं मिल पाया।
और इस टाइम में कुछ प्लानिंग है क्या?
अभी तो नहीं, उधर से भी निकल गए सारे लोग, मेरे इन लॉज भी निकल गए, बड़ौदा ट्रांसफर चले गए।
फिर स्कूल कौन चला रहा?
वो मेरा कजिन है, मेरा ये ब्रदर इन लॉ है, वो
ओके
थोड़ा बहुत रहता है।
ओके 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 ओके।
नीता किसको ये बनाना है होस्ट?
क्या बोले?
नहीं नहीं नहीं, नीता को पूछ रहा था होस्ट, मिलसेटा को होस्ट। नीता को सूचना। 10 मिनट में हम चालू करेंगे शेख।
ओके ओके प्रवीण जी, नो
10
प्रॉब्लम।
मिनट में 4 बजे का टाइम दिया था।
ओके नो प्रॉब्लम प्रवीण जी।
या और बोलिए, स्टूडेंट्स आ रहे हैं अभी। एक फाइनल रिमाइंडर दे रहा हूँ सबको। भाभी कैसी है? शेख भाई
अच्छे, वो भी जॉब कर रहे हैं अभी।
किधर कर रहे हैं?
वो गवर्नमेंट में एनआरएचएम, नेशनल रूरल हेल्थ प्रोग्राम के अंडर जो पोजीशंस भरे थे ना,
हाँ,
उसमें जॉब कर रहे हैं वो।
हमारा कॉलेज भी अभी एनएसएस के लिए सेलेक्ट हो गया था, हम भी ये काम कर रहे हैं अभी। न्यूट्रिशन और इम्यूनाइजेशन के ऊपर यूनिसेफ का प्रोग्राम के लिए हम लोग सेलेक्ट हो गए कॉलेज।
कौन सा
सो
बोले प्रमोद जी?
यूनिसेफ के प्रोग्राम है एक, ये एनएसएस का,
हाँ, एनएसएस का।
इम्यूनाइजेशन एंड न्यूट्रिशन।
हाँ।
तो हमारा कॉलेज यूनिसेफ का प्रोग्राम में सेलेक्ट हुआ है।
अरे वाह वाह, बहुत अच्छा, वेरी गुड।
तो अभी डब्ल्यूएचओ का कंसल्टेंट परसों आ रहे हैं हमारे कॉलेज। सो
हाँ हाँ।
भी प्रेजेंटिंग, प्लस हमारे लोकल पीएचसी के वो भी आ रहे हैं।
अरे वाह, वेरी गुड।
एंड एक लोकल जो नेटवर्क है,
हाँ।
वो भी, उनको भी हमने इनवाइट किया है प्रोग्राम के लिए।
अरे वाह।
तो ट्रेनिंग हमारा यूनिसेफ के एक मेन ट्रेनर के अंडर हुआ था, मेरा 3 दिन का हुआ था एनएसएस की
हाँ
तरफ से।
हाँ हाँ।
तो वो हम कर रहे हैं।
राष्ट्रीय बाल स्वास्थ्य कार्यक्रम है ना,
हाँ 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 हाँ।
उसमें है वाइफ में हमारा।
ओके ओके ओके, बिकॉज ये भी उसमें ही
नेशनल।
आता है, न्यूट्रिशन में
हाँ
ही आता है। न्यूट्रिशन और इम्यूनाइजेशन में ही आता है वो भी।
हाँ हाँ, बराबर, करेक्ट है, करेक्ट है। आपका कैसे चल रहा है प्रमोद जी?
बस चल रहा है, बच्चों को पढ़ा रहा हूँ केमिस्ट्री।
अरे वाह वाह। ये अलोका फाउंडेशन और
हाँ,
कॉलेज एक ही है, अलग अलग है?
अलग 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 अलग
ये
है।
अभी प्रोग्राम अलोका फाउंडेशन के
और
थ्रू
आलो
पे?
के फाउंडेशन की तरफ से हो रहा है।
ओके ओके, और आप
हाँ,
कौन से कॉलेज में?
मैं श्रीदेवी कॉलेज ऑफ फार्मेसी में आपको होस्ट बनाता हूँ। कौन सा वाला इसको होस्ट करो शेख भाई?
हाँ
आप जो बात कर रहे हो उसको मैं आपको होस्ट बनाता हूँ, ताकि आपको शेयर करना पड़ेगा ना।
हाँ हाँ, ठीक है, इसको, ये जो बात कर रहा हूँ इसको होस्ट
हाँ
बनाइए।
हाँ, मैंने कर दिया सर आपके।
ओके।
हाँ, और एक है
करो स्क्रीन। हाँ।
वो भी दूसरे, उसको भी मैं आपको ये कर देता हूँ लाइव। हम लोग यूट्यूब में भी लाइव है शेख भाई।
ओके
वी आर लाइव ऑन यूट्यूब ऑल्सो।
ओके ओके। नो।
सो तो दोनों के, अरे आपको गलती से होस्ट बना दिया मैंने। हाँ, आप मेरे को को होस्ट होस्ट बना दो शेख भाई, नहीं तो मैं इधर से कंट्रोल नहीं कर पाऊंगा। को होस्ट की जगह पे होस्ट कर दिया मैंने आपको। आप सेटिंग में राइट क्लिक में जाके मेरा इसमें, हाँ, नीता, no, Nita, you are the host now. You please make me host. Nita, can you hear me?
Yes, sir.
Can you make me host? Because I think by mistake it has happened. You can make me host. Yeah, I will just. Thank you.
Yes, sir.
Nita, I'll also make you host. Host. Nita. So it's another 5 minutes. Yeah, it's 3:51. I think by 5 minutes people will be joining us. So I request all of you to invite your friends. I've shared the link. Please join for the session.
communicating effectively. With Grammarly,
Meet Ty. He is emailing a difficult client.
I think we should
Organization. We were happy to receive abundance of love and donation, which helped us to help the Sevashram organization.
First event of our organization post the logo launch was a Go Green Tree Plantation Drive wherein the members distributed around 50 saplings to all the prominent people in Mangalore, students and various institutions to promote the importance of planting new life.
COVID-19 in 2020 was not just a huge challenge for us humans but also animals. The generous donations received by the donors to our foundation helped us to conduct a cattle feed drive at Kapila Park Goshala, an animal protection organization at Kenchar, Mangalore, helping the cattle with the food requirements. Also, few members from our team spent a
to care for just like you and me. Keeping this in mind, Aloka Foundation collaborated with the very well-known Animal Care Trust in Shaktinagara, Mangalore to supply various essentials to the animals shelter. Along with this, our foundation also conducted an adoption drive by preparing pamphlets for the same. We were successful in helping few of these adorable ones to get homes.
During the times of pandemic, it is important for people to know the right method to stay protected. The members of Aloka Foundation distributed COVID-19 awareness pamphlets to various places including grocery stores, organizations, other institutions and public health centers across Bangalore to spread the right information on staying safe from COVID-19.
The best part of being a human is about helping others. In these difficult times of pandemic, Aloka Foundation and its team reached out to people and few families and helped them by supplying food and daily necessities.
Giving is not just about making a donation, it is about making a difference. Our organization conducted a stationery drive where the funds were used to gather the stationery items for the orphanage students. The success of this event could be seen in the smiles of these kids who are the future of our country.
A beautiful woman draws strength from troubles, smiles during this distress, and grows stronger with prayer and hope. Aloka Foundation was fortunate enough to celebrate this year's Women's Day on 8th March 2021 at Sevashram organization at Tellakatte, Mangalore. A fundraiser camping was conducted a week before the Women's Day in order to
Can you hear the video, everybody?
present in the Sevashram organization. We were happy to receive abundance of love and donation, which helped us to help the Sevashram organization with food and other daily necessities.
Though this pandemic made us shift our academics and seminars from classrooms and seminar halls to computer or mobile screens, this did not stop the process of learning. Aloka Foundation, in collaboration with many educationists and speakers across the country, conducted free online webinars for students and learning enthusiasts on topics that benefit students and the topics related to health.
These competitions were conducted for raising awareness as well as raising funds for certain events like Women's Day, mental health awareness, COVID-19 relief funds, and many more. Make sure you follow us on YouTube, Instagram, Twitter, and Facebook to participate in these competitions.
A heartfelt gratitude to all our supporters. Thank you for watching. Make sure you share, subscribe and like this video. And
Thank you. Thank you all. I would request Vilsita to start with the program with our introduction. I'll share.
Yes, sir.
I'll share the screen. Just a minute. Can everybody see the screen? Vilsita, you can start.
Yes, sir. Before we start, I request everyone to keep yourself on mute. Good evening, everybody. I, Ms. Vilsita, welcome you all today to the third webinar conducted by Aloka Foundation, Mangalore. I welcome all the participants to this virtual webinar series on today's topic of discussion, role of Indian pharmaceutical companies in the development of new drug molecules. I also take this opportunity to welcome the founder members of Aloka Foundation who are responsible for the progressive growth of this foundation. Our President, Professor Pramod Bhaskar Kumar, Secretary, Mrs. Rihab Tonse, and Treasurer, Mrs. Kantini Pramod Kumar to this event. Before we introduce our guest speaker of the evening, let me take this opportunity to say a few words about our foundation and the activities we do. Aloka Foundation came into existence on 6th September 2021, starting off with few handful of students and Mr. Pramod Kumar. Above anything else, the main vision of this little non-profit foundation is to help and to do all sorts of humanitarian work. As Anne Frank once mentioned in her quote, "No one has ever become poor by giving." The members and volunteers associated with this foundation selflessly work round the clock planning different activities and events that can help people, animals, and even environment in one way or the other. From a team of just 15 members in the beginning, our organization has now grown into a team of 60 plus members and many volunteers from different states across. Nothing is easy at the beginning, but the team members never lost hope. Under the guidance of the founder members, we started conducting various events and drives for the betterment of the society, and are proud to say that we have coordinated with some of the most esteemed institutions across Mangalore for conducting health awareness drives and fundraising events. We have also coordinated with the Animal Care Trust Mangalore during pet adoption drives and donated essentials to the sheltered animals. Our team have conducted various donation drives in organizations across Mangalore like Snehalaya Trust, Sevashrama, Kapila Goshala, and many more. Our members conducted plantation drive across Mangalore on the occasion of World Health Day, World Earth Day. Not only that, in this monotonous age of mobiles and gadgets, we have managed to take way into people's social feed by giving daily updates about various important days and various awareness videos throughout the time of pandemic and continue to do so. We believe that this is just the beginning as we have many more people to help and many more dreams to achieve to make the society a better place to live. It is my distinct privilege and honor to introduce today's guest speaker. He is a very kind, hardworking human being who has accomplished immense amount of success in his field. I am pleased to welcome Dr. Mohammad Usman Sheikh to today's webinar on behalf of our foundation. He completed his PhD in organic chemistry from Dr. Baba Saheb Ambedkar Marathwada University, Aurangabad. With over 20 years exposure in new drug discovery, he also served as a group leader
in Wokhart Research Center and was also a part of Suven Life Sciences Limited and Harman Pinochem Limited as research chemist. His academic research papers include synthesis and characterization of some biologically important triazoles and synthesis of diacetates on silica gel surface. He holds total 5 patents and has also published his work in various journals in the span of 2006 to 2018. He was also a part of various workshops and conferences such as International Conference of Organic Synthesis, 51st Annual Conventions of Chemists, 10th ISCB International Conference on Drug Discovery, Perspective and Challenge, 64th Indian Pharmaceutical Congress, etc. We welcome you to this webinar again, sir. Now, without any further delay, I request our speaker to take over this session. Thank you.
Dr. Mohammad Sheikh, you can start with your presentation, sir.
Very good evening to all of you.
Mohammed sir, are you finding any difficulty in sharing the screen?
No.
Okay. We can't see your screen as such.
Wait, Mujia.
Okay.
Just a minute.
Okay, sure, sir. Thank you. The screen is visible, Sheikh sir.
Very good evening to all of you. Thank you, Vilsita, for nice introduction.
Sir, can you go for full screen? Sheikh sir, can we go for full screen, sir?
Just a minute, huh? Yes. Hello. Hello.
Yeah, tell me. It's not on a full screen mode. We can see your screen. Can you just put it in full screen mode, sir?
Now I have shared full screen. Are you getting?
I am not. Can anybody sit? No. Yeah, you can carry on, sir. You can go ahead. Yeah.
Just a minute.
Excuse me.
Just a minute. Huh?
Should I share it for you? If you can share, and the PPT, I'll share it. I'll do. Yeah, now. Yeah. Now it's full screen. Now it's full screen.
Okay.
Yes. Thank you.
Okay, thank you much. Good evening to all of you. Today I am here to present role of Indian pharmaceutical companies in the development of new drug molecule. I am thankful to Aloka Foundation for giving me this opportunity, and especially thank to Mr. Pramod Kumar for giving me this opportunity. I am working with Pramod from 3 years, 2005 to 2007 in new drug discovery department. Right now, I am having 20 years experience in Oak Research Center in new drug discovery medicinal chemistry department. My presentation contains introduction to drug discovery and development, role of Indian pharmaceutical companies, and conclusion. What is drug? A chemical substance of known structure other than nutrient or an essential dietary ingredient which when administered to a living organism produces a biological effect. Any substances or product that is used to intend to be used to modify or explore the psychological system or pathological state in the benefit of the recipient. Simply, a substance used as medicine is drug. Any substances or product that is used in the treatment, cure, prevention or diagnosis of disease or used to otherwise enhance physical or mental well-being. What is branded drug or generic drug? Branded drug, also known as innovator drug. Branded drug is the product that is patented by inventor company, can be produced and sold by the company holding patent, costly to cover the R&D expenses. Then generic drug, comparable to the branded drug in the dosage form, strength, route of administration, quality, performance, and intended use, can be produced and sold after original patent expire. Cheaper, less development cost. Drug discovery program, synthesis and biological screening of compounds. Then it takes lead optimization. After lead optimization, drug development or candidate development, that is chirality, salt formation, and polymorphs. Then the compound enter in preclinical studies. In between preclinical studies and IND and clinical development, failure due to non-drug-like properties, so that the major step of the compound development is preclinical study and then IND and clinical development process. What is drug discovery? The process through which potential new medicines are identified. It involves a wide range of scientific disciplines, including biology, chemistry, and pharmacology. In other way, the process by which new candidate modifications are discovered once a compound that fulfill all of these requirements has been identified and the process of development continues. There are 3 stages of drug discovery and synthesis. First stage, drug discovery. Drug discovery is nothing. Candidate molecules are chosen on the basis of their pharmacological properties. Then second stage is preclinical development. Non-human studies, toxicity testing, pharmacokinetic analysis, and formulation development are performed. Clinical development, third stage. The selected compound is tested for efficacy, side effects, and potential dangers in volunteers and patients. Drug discovery process having 5 main stages. Stage 1, discovery and development. Stage 2, preclinical research. Stage 3, clinical development. Stage 4, FDA review. And final and most important stage is marketing. History of drug discovery. History of drug discovery. In pre-1919, herbal drugs are discovered, and then serendipitous discoveries are there. In 1920s and 30s, vitamins and vaccines are discovered. In 1940s, antibiotic era is there, and in 1940s also R&D boost due to World War II. Then in 1950s, new technology and discovery of DNA. In 1960s, breakthrough in etiology. In 1970s, rise of biotechnology and use of information technology. 1980s, commercialization of discovery and combinatorial chemistry. In 1990s, robotic and atomization takes place. Total drug discovery program. Total drug discovery program. Drug discovery, the target selection, lead finding, lead optimization, pharmacological profiling. It will take around 2 to 5 years, and 100 projects are started for drug discovery program. Then total around 20 compounds were screened and entered in preclinical development. In preclinical development, pharmacokinetics, short-term toxicology, formulation, and synthesis and scale-up of the molecules. In between drug discovery and preclinical development, the drug candidates were selected. And then after preclinical development, the drug development compound was entered in clinical development, a very important stage. And almost 10 compounds were entered in clinical development. Phase 1, phase 2, phase 3. Phase 1 studies pharmacokinetics, tolerability, side effects in healthy volunteers. Phase 2, small scale trials in patients to assess efficacy and dosage. Long-term toxicology studies. It will take around 5 to 7 years, and almost 1-2 molecule were entered in regulatory submission. Regulatory approval, submission of full date and review by the regulatory agencies. It will take around 1 to 2 years, and one molecule entered in phase 4, post why R&D cost is high. Almost 10,000 drug candidates were selected, but the compound which was going for a drug molecule should be potent, selective, reversible, targeted, soluble, long-lasting, metabolically stable, and permeable. Main and important stage is toxicity. The molecule should be non-toxic, non-mutagenic, non-teratogenic, physically stable, patentable, and manufacturable. The challenges, these are the challenges of drug discovery, finding one molecule that meets multiple criteria. desirable profile of NC. Profile of ONC dependent on integrity, permeability, API stability, CYP 450 inhibition, major metabolic stability, blood-brain barrier, PK, PD studies, log P solubility. These are the profile of NC. Further test for NC to become drug. What is drug? It in vitro efficacy in animal, pharmacokinetic and pharmacodynamic studies, safety in animals, formulation studies, levels in healthy subjects P1, efficacy in patients P2, ethnic group P3. These are the major factors having drug. Drug discovery, total drug discovery program. First, the chemical synthesis or NC synthesis, it will enter in vitro and in vivo screening, that is lead optimization. Then the compound enter in preclinical toxicology. This phase is nothing but multidisciplinary discovery phase. It will take around 3 to 4 years and cost around 20%. Then before IND filing, the applied research, that is analytical studies, scale up and development of the drug, then manufacturing process and dosage form. Then compounds enter in clinical trials, that is phase 1, phase 2, phase 3. Clinical research takes around 3 to 6 years and cost around 55%. Applied research cost around 25%. Then the compounds enters in FDA review of NDA, that is regulatory market. It will take around 1 to 2 years. And finally, compounds enter in market. Estimated international drug discovery cost around $300 to $500 million. Reason for failure of a drug. There are many reasons. Because of commercial reasons, 5% the drug fails. Animal toxicity 11%, adverse effect in human around 10%, lack of efficacy of drug 30%, poor pharmaceutical properties around 39%. The main part of the presentation is role of Indian pharmaceutical companies. Drug discovery setups in India, complete drug discovery setups are computational chemistry, medicinal chemistry, analytical development, process development, biological setup, PK/PD studies, toxicology study, clinical research, and regulatory. Complete setup having Wockhardt, Dr. Reddy's, Ranbaxy, Zydus Cadila, Glenmark, and Orchid Pharma. Custom research organizations are computational, Schrödinger, medicinal chemistry, Adventinium, analytical, Vimta Labs, process Almino, biological toxicology study, Advinus, and clinical research Sigma. Business model in India. Develop drug on its own basis. Development of new chemical entity to the market level, total risk is taken by the company, Wockhardt, Zydus Cadila. Collaborated with multinational companies. Development of new chemical entity to the preclinical or phase 1 and collaborate with multinational company for further development, risk sharing with multinational company, that is Glenmark, Reddy's, Orchid Pharma. Part knowledge-based service providers confined to the developmental service as required, Adventinium, Advinus, GVK Life Sciences, and Vimta. Scenario in drug discovery. Work in already established therapeutic area as development guidelines and liabilities are known. mostly work on weaknesses and advantages over competitors' drug in the market for the product to be superior somewhere. That is spectrum, toxicity, oral, OD. Classical structural activity relationship development guided often by computational means. Mostly dependent or quicker in vitro test to the screen compounds. Compromises on fail early decisions due to pressure to perform. Less culture of indisciplinary research. History of Wockhart's drug discovery. Wockhart started antibiotic drug discovery in 1997. Around 25 years of focus on drug discovery research. 145 plus scientists, 43 of these are PhDs. Comprehensive drug discovery capabilities. Identified drug candidates from 4 different classes of antibiotic. Result of 20 years of Wockhart antibiotic discovery program. More than 145 discovery team. More than 7,500 molecules were synthesized. In 20 years, 5 drugs at phase 3 stage. More than 80% of success rate. Wockhart antibiotic addressing AMR. That is, there are 5 drugs: WCK4282, hospital infections, WCK5222, as well as WCK771 and WCK2349. That is nothing but drugs in Indian market as AMROC, community infections, as well as WCK4873. Wockhart antibiotic recognition by the global health authorities. All 5 Wockhart antibiotics accorded with QIDP, qualified infectious disease product, and prominently covered in WHO report. Wockhart antibiotic pharma industry in India. The pharmaceutical industry in India is more stable compared to the other industries, and it is expected to continue so with impressive performance. Small scale enterprises, small scale and medium scale enterprises are also likely to play a substantial role in the growth of the India pharma sector. According to consulting firm Grant Thornton, the country will also see the largest number of merger and acquisition in the pharmaceutical and healthcare sector in coming years. A quote from Richard Grester: "The Indian pharmaceutical industry is a success story, providing employment for millions and ensuring that essential drugs at affordable prices are available to the vast population of this subcontinent." Conclusion: drug discovery process is tedious and expensive, where the drug discovery process is tedious and expensive, where the risk of failure is more. More of the Indian origin drugs are herbal medicines, where research expenses are less compared to the synthetic drugs. Need to train discovery people from beginning if results are required. Need to develop interdisciplinary team, and training also required for an individual to become a team player. Next 10 years are crucial for Indian drug discovery. Researchers as many molecules under final stages and development in many Indian companies will decide the path to go forward or backward. Thank you so much. Any questions?
Thank you, Dr. Sheikh. Being a part with you in our drug discovery program, where we have done a lot of research during our time. Yeah, we have done a lot of research, and as rightly said by you during your presentation, that a molecule becomes a drug. It has to pass through various phases. It has to pass through various phases of drug discovery. So, and as we have done, as rightly explained by you, that during this process, there are a lot of challenges. It is financial challenges, it's molecular challenges, whether even after doing so many research, getting so many molecules, as we have shown that number of molecules synthesized and number of actives that we have got is very less. So drug discovery process, as very well explained by you, is not a simple thing. If in every phase there are challenges. In every phase, can you hear me?
Yeah, bro.
Yeah, there are many challenges that we come across when we do drug discovery. When we do it, as we were working on one of the modification of macrolides. I was working with Dr. Milind. We are working on macrolides, and Dr. Patil's we were working on antimicrobial activity, which was similar to linolazide.
Correct. Oxazolidinone class.
Yeah, that we were doing it. So it was one of the most challenging things, and to get that particular configuration and isomer was the great thing. So chemical stability, as well said, that okay, everything is achieved at the end of the day. If you are not able to get the desired product with some changes, it do hinders our drug discovery program.
Yes.
So that's the most important thing in what we do in colleges. What we do in colleges, it is just a small part. We just design small modification, but when we do a real drug discovery, when real drug discovery goes with there, we do a lot of modification. We see ifs and buts are seen. Your computational study comes into play. We do the docking study, and after having a lot of brainstorming sessions, we design a drug. So a drug coming out from a drug discovery process, it's a long way. It's a huge tunnel where the end, we see that today it's the end comes, but somewhere down the line there will be some leakages which has to be filled. So drug discovery is a long process, as well as very well explained by Dr. Sheikh. And being a part of drug discovery, it's a patience. Being a part of drug discovery, it's a really patient work, where we have to think, okay, today we think that we have got one molecule. And I'm glad that I worked with Dr. Sheikh with one of the molecules with Dr. Patil. We were working on one molecule.
You are in scale up, Pramod.
Yeah.
You are in scale up and
scale up. Yeah, I still remember.
And development of that molecule.
Yeah, it was one of the most active. And that was preparation, and we did a lot of research. The process was set, but to make it scalable was one of the most difficult thing. Molecule making in 1 gram, 1 mg is okay, but to scale it up, it's a big job. People think that 1 mg making it's okay, but to scale it up to a higher level for the production, for animal studies and all, it is grams, kilograms that we have to make. So that is a real challenge, and that's where the chemist, a medicinal chemist, plays an important role. His expertise in the field, his knowledge on the work that has been done, small reaction modification, the cost reduction. That's the most important thing in drug discovery. There's a huge cost when we go for production on higher scale, 1 mg, 2 mg, 10 mg initial screening. Yeah, it does not matter much. But when we go on a scale up mode, when the drug becomes active, so that's where most important part comes. So we in a team when I was working with Dr. Sheikh, and I can still remember Dr. Shivaji Pawar. There are many Deepak Dake. So Dekne, sorry. So we were really lucky enough to have a good teamwork where sharing of knowledge, and most important, as every time I say, sharing of knowledge, looking out for what is going wrong, because a good medicinal chemistry looks out for a small thing. Sometimes small reactions goes hayward. So that all things is not. So in drug discovery process, it's a long process where we have to be vigilant. As said by this thing, there are challenges are there in each and every step, right from synthetic to marketing. We have to follow various steps, and that was very well said by Dr. Sheikh. And I'm glad that you could interact with my students. And my students, I just want you, see, those who aim in going to be in part of a research organization, you can put on your questions to Dr. Sheikh, how you can be a part of it. So if there is any queries, you can put it on the chat box, or you can ask your questions. This thing, and yeah, kindly fill the feedbacks. The link is provided about the session. And if you have any queries, please do ask our guest speaker, Dr. Sheikh, Muhammad Sheikh. So if there is any doubts, yeah, the session is open for question answers. Anybody wants to have any doubts? I think there are some chat box. I'll just check it up if there is anything over there. So yeah, as usually people are very reluctant in asking questions. Huh, Sheikh? Yes, most of the time people are very reluctant in asking questions. So, but I request everybody to be more vocal. Let the question be silly, as every time I say. Excuse me. Every question is a question. Every question is a, yeah, we will send a, every question is a question. So yeah, I think today Dr. Vijay Kumar Aradhya sir from Shimoga has joined us. He is the senior professor over there in Shimoga National Medical College of Pharmacy. So it's glad that sir has joined us. So any more doubts, please do come and ask. And I'm thankful to Dr. Sheikh for sparing his time from his schedule. And Eid Mubarak, sir. Abhi last week mein Eid hua. Eid Mubarak to you, sir.
Hello.
Yeah, Sheikh, sir, can you hear me?
Hi, yeah.
Yeah. So would you like to share some experience of your during your drug discovery process? What all challenges you faced? Since we have done it with this, can you just any molecule that you've done? Okay, that problem, because people think that drug discovery is just a streamlined process. So can you just share some of your experience? You use 20 years experience. Can you just share about you?
Main task when the compound was linear and multi-step, then the hurdle was to synthesize compound. We shall plan a proper route that is workable. And most important thing is that if any of coupling or rearrangement, some step creating some problem, then we have to change the route in non-infringing route, and that route should be workable. Compound should be isolable and pure. One more task is that purification technology. Some compounds are highly polar, then it should be recrystallized, not purified by column chromatography or combi flash or other techniques. That is also very challenging. One most and focused thing is that route selection is both very important in synthetic chemistry, organic chemistry. And when we start synthesis, we have to synthesize compound in a systematic route, not in many ways. We have to select a perfect route and then start synthesis, and backup material also ready for the compound synthesis up to the final stage. When we get compound and the compound is active, the most challenging task is scale up, because in scale up process, column purification in gram scale is very difficult. Then we have to choose route in that way that compound should be purified by crystallization technology, not by column chromatography, because in column chromatography solvents are used in huge quantity and scale up takes more time. Then time also very important. Reagent or isolations, target, all these things are considered and focus. That if you are having multi-step process, then we have to choose a team and started work in this fashion, that side chain will prepare some work somewhere, and final stage we have to couple in that fashion that compound gets isolated in short time. And main selection of route is that protection and deprotection of the groups. That is very important. If you protect NH by Bock, then we have to use acid to deprotect it. If you use benzyl protection, then we have to debenzylate using catalyst. That is very important. We have to think of scale up process also, not only synthesis, but in scale up also it is very important. Choosing of reagents or choosing of synthetic scheme, analytical support is very important, and the NMR, mass, HPLC, C13, all these analytical tools, as well as purification technology. Main and important thing is that compound isolation. Some compounds were highly polar. They are having polar functional group, that is sulfonic acid or amino groups, that will isolate by making salt formation.
As rightly said by Dr. Sheikh, because as scalability is one of the most important thing, because we were working on one reduction process in that acetate. Oxazolidone synthesis, there was a reduction process. So there was palladium carbon was used in a huge reactor, which was very difficult. Using palladium, it's highly explosive. So we were highly explosive. Handling palladium carbon is not that easy. And I still remember once we were doing one batch, it had caught, while removing the reactor, it had caught fire. So it's very challenging. So we have to look out for an alternate way where such exposures using raninical, where there is possibility of fire catch and all. So we have to look out for an alternate reducing agent. Yeah, it might be time consuming, but yeah, when you go for a scale up batch, all these factors and the cost. So yeah, as rightly said, and the most important thing, once the drug gets active, once the drug is active, the most part comes after that. Once you have done the animal studies, the metabolite study is the most important thing. We have to synthesize the metabolite. The metabolite will be isolated by the DMPK team. They will tell us to synthesize what the body drugs to the drug. We have to, as a chemist, we have to synthesize it to be analyzed. That's the most challenging part in drug discovery, what I feel, because when I was working in Torrent Research Center, I just wanted to share with this with my students. We are one of the most active drug in metabolic disorder. They were doing human studies and they found some metabolite. And after doing LCMS study and all, they could know exactly what the molecule was. The challenge is what body drugs to the drug. To synthesize it in lab is the most challenging thing. And that's where the most challenging part comes in our in the life of a chemist. Whenever a medicinal chemist, when he works, when it goes to such a level where the compound will be even 1% of an impure metabolite, if it provides it creates toxicity, then it is a problem.
Correct.
So that's the most important thing that you have to know. And as I would like to tell you that there is a special branch in drug discovery, which is impurity profiling, which is impurity profiling, where people just synthesize impurities and get lakhs of rupees. Get lakhs of rupees, because that impurities determine the purity of the camp. That determines the purity of the sample. So anybody has any doubts? Because see, it is a vast process. It's a vast process. It's a vast process where we keep on working on it. It's not a one-day thing that you get it. So I would like to say that if you're planning to be a scientist, and I'm glad to work in research for 13 years with eminent scientists like Dr. Sheikh. There are many more, Dr. Patil, Dr. Deshpande, Prasad Deshpande, to name few very innovative problem-solving people. It is not synthesizing a compound. Synthesizing a compound is very in mgs. Okay, you can do it. But the main factor comes when you do the scale up, as rightly said by sir. 1 mg, 5 mg you can prepare, but when the compound becomes active, then your real challenge comes. And rightly said, the techniques of the knowledge of a medicinal chemist plays a very important role when you go for a scale up. So I'm thankful to, and I'm glad that Suvarna Kini ma'am has joined the session. Thank you, ma'am. She's the HOD for Manipal College of Pharmacy, Pharmaceutical Sciences, Manipal. My guide also for MPharm. I'm glad that she has taken her time out to be a part of this webinar series. She has been frequently joining us. Thank you, ma'am, for your encouragement by joining these sessions.
I hope I'm not late.
No, ma'am. Anytime, because you can still refer this. This presentation is live on YouTube also. Anytime you can.
Okay.
You can. It is there on YouTube. You can go to YouTube Aloka. You can get this. You can anytime refer the webinars because it is there live. So anything, if you have anybody who have missed because of, there are some prior commitments for everybody. Yeah, that's why we are always available on YouTube, so that even if you miss something, yeah, if the topic is interesting, you can always go back and refer to YouTube. And I'm thankful to all the participants who have come down over here, sparing their time on a Sunday afternoon. Yeah, most of the webinars are on Sunday afternoons. Yeah, sleeping time, rest time, but still coming back down over here, listening to our speakers. We are grateful to all of them who have been a part of this webinar series. Yeah, in future, yeah, I'm just in coordination with some of the speakers right now. Once I get the confirmation, yeah, surely we'll get back to you of the future webinars. And yeah, it should be something that will generate interest in people. That's our main idea in doing these webinars. So if there is any doubt, you can please ask. And I'm thankful to Dr. Mohammad Sheikh for accepting our invitation to deliver a webinar on a topic where it relates the academics to it gives a knowledge to our students. What are their roles in drug discovery?
Thank you, Pramod, and all the best. Aloka Foundation having wonderful quote that opening minds, changing lives. Great work. I have seen your video, the great work you have done through Aloka Foundation, and all the best, Pramod, and thank you so much, Aloka Foundation, for giving me this opportunity. Thank you.
Thank you. I would like to now Vilsita to give the vote of thanks for the session.
Now I would like to call upon Ms. Cheryl Pinto to deliver vote of thanks.
Thank you, Vilsita. At times, our own light goes out and is rekindled by a spark from another person. Each of us has cause to think with deep gratitude of those who have lighted the flame within us. I'm sure today's webinar has rekindled in all of us the spark of wanting to learn more. Good evening to all the participants and members attending today's webinar. As we come to the end of today's session, on behalf of Aloka Foundation Mangla, I deem the honor and privilege to deliver the vote of thanks. Let me first start by showing my sincere appreciation to our guest speaker for today, Dr. Muhammad Usman Sheikh, for taking out time of his busy schedule to address an important topic. I'm sure that we all have learned some new things today and are taking a lot of knowledge about this topic with us. Thank you once again, sir. I would also like to thank Mr. Pramod Kumar, President, Aloka Foundation Mangalore, for his never-ending support during the event. I owe special gratitude to all the members of the foundation for their time and effort in making all things fall in place. Lastly, I would like to extend a big thank you to all the participants who have attended today's webinar, without whom the success of this webinar was not possible. Thank you all. Reminder, for those who have not filled the feedback forms, kindly click on the link in the chat box. Thank you.
Thank you.
Thank you, Cheryl, for kind words. Yeah. And in Aloka, yeah, we strive hard to help out people. Huh? Yeah, Mr. Nandish, yeah, you can, anybody, can you just put up the link again, somebody in the chat box, so that people who have not filled it, yeah, it's there, Mr. Nandish, you can fill the feedback form. And I'm thankful again, Sheikh, Dr. Mohammad Sheikh. I think he is a very good, I'll tell you one good thing about Sheikh, Mr. Mohammad Sheikh. He is a very good, क्या बोलते हैं आप शायरी बहुत अच्छे से करते हो, and I get inspired by his शायरी daily status update. It's very inspiring when I daily I make it a point to see his status, because it's very inspiring. The जो आप शायरी डालते हो बहुत मायने रखते हैं जिंदगी में हर एक के.
Thank you.
I'm really grateful, because it creates a positive thinking in our mind. So, and I always look out the day that, okay, I just see for your, आज भी बहुत अच्छा आपने एक Mother's Day, and I wish every woman, mother, a happy Mother's Day. And mothers are the most important thing in anybody's life. They are the one who make us. Let it be anybody. जो एक बहुत ही अच्छा मैसेज आज आपने स्टेटस में डाला था, मैं जो सुन रहा था एक किसी, so it's really inspiring that I feel that. And by today's Mother's Day, and yeah, I do miss my mom. Yeah, so they are the pillars. Yeah, Atulya, you want to say something? Yeah, so yeah, we do miss, I do miss my mom most, and she has been the inspiration for me in doing many things in my life. Whatever work I take, I keep her in my mind and do it. So somewhere, wherever she keeps, I get my blessings and support wherever she is from her. So grateful, because mothers are the most important thing in life, because father can earn, give a living to you, but mothers bring up the best thing from you. They are the support. I think without mothers, I don't have anything to say. Basically, they are the backbone of the house. हम कितना भी कमाएं, जब तक माँ घर में नहीं हो, उस घर को घर नहीं बता सकता. So the women in our house who take care of our kids are the most important thing in our life. So I want everybody to respect the woman in your house. So let it be your brother, sister, wife, anybody. Mother is the most important thing. Respect them. And with that, I would like to thank all of them who have been part, and wishing all the mothers a happy Mother's Day. And their sacrifices, yeah, not today, it is endless. It is endless. So I thank all of you again for participating in this webinar session. Thank you, Dr. Mohammad Sheikh, for accepting our webinar. So with this, I would like to end this session. to all and thanking you everyone for your attendance. Good day. Thank you all.